Dr. Ophelia Venturelli, who's assistant professor at the University of Wisconsin-Madison. Ophelia received her PhD at the California Institute of Technology, and she studies spatiotemporal behavior of microbial communities with a goal to manipulate and engineer them. In today's talk, Ophelia will tell us about predicting and designing community-level functions of the human gut microbiome. Ophelia?
Wanted to thank the organizers for the opportunity to come visit Berkeley. It's been a little while. I did my postdoc with Adam Markin a few years ago, I guess more than a few now. So I'm excited to tell you about some of the work that we're doing in my group. We are interested in how to design synthetic communities from the bottom up and understand the principles by which they function and new control knobs for manipulation. So microbes have evolved in microbial communities. They didn't evolve in isolation, but we've been studying them in isolation for a very long time. Now we're starting to embrace the complexity of these systems. But in order to actually understand these communities and how microbes function, we have to understand the interactions that are within these communities and shaping their growth and metabolic activities. So in microbial communities, microbes' functions are expanded and enabled. They also exhibit greater stability, and they can utilize a wider range of resources in the environment. So a lot of reasons why we want to study and engineer microbial communities. And their applications can be harnessed for a variety of applications in human health, agriculture, bioprocessing, environmental cleanup. So the sky's the limit in terms of what we can do with microbiomes. So our group is interested in a kind of reductionist approach for studying microbiomes. We build communities from the bottom up. We reduce the complexity of these natural systems that have hundreds to thousands of different microbes, and then we can actually control who's in the community. We use high throughput approaches for studying microbial communities, and so we have a toolbox of different microbes. We can combine them into communities and use a variety of different platforms for studying their function. In vivo, in germ-free animals, we also do work to study the human gut microbiome and its interactions with the host. And then we also have high throughput approaches using liquid handling robots and also droplet microfluidics, which allows us to measure 1 million different communities simultaneously or single microbes. And for some reason this video, oops, not playing, but yeah, so we can encapsulate microbial communities into droplets and analyze their properties, and those are ongoing work in our group. We're also interested in single cell heterogeneity within these communities, and so we've started to look at the genes that are in individual microbes, and we've used this to study phase variation and antibiotic resistance genes that move through a community. Unfortunately, I'm not going to have time to talk about some of those things today, but I'm going to share some of the work that we're doing with our human gut communities. So microbiomes microbes live in close proximity in microbiomes, and they're constantly interacting through a variety of different mechanisms. We don't actually know the interactions. So one of our interests is how to map those interactions quantitatively and decipher them, and then exploit those mechanisms for control. These interactions range from resource competition to biomolecular warfare through the release of antimicrobials. Microbes also produce metabolites that other microbes can use. And this collectively as a unit generates the outputs of these communities. We're interested in metabolites and how to harness these metabolic capabilities. But a key challenge is also the stability and resilience of communities, because if we can engineer them to do a specific metabolic function, that's one step forward. But if we can't get them to perform their function over a given time frame, then we're pretty much not able to have an effective microbiome engineering solution. And so stability and resilience to perturbations is really critical for thinking about how to design microbiomes for our benefit. And one of the biggest challenges with studying microbiomes is the immense complexity. There are hundreds to thousands of different microbes in these communities. And so the number of possible communities that we can study in the lab using our bottom-up approaches scales exponentially with the number of species that we have or environmental factors that we're considering. And so this creates a huge design landscape that we have to navigate efficiently. So we combine these high throughput approaches with computational models and use our models and experiments designed together to further our understanding of microbiomes so that we can engineer them. Also, another challenge is a change as a function of time and space. So they're very dynamic systems, and so we have to consider those dynamics when we're thinking about engineering and understanding what these microbes do. So I'm going to tell kind of two vignettes from our group, focus more on the human health side. So we've been interested in the human gut microbiome for quite some time, and it's a teeming environment teeming with microbes, highly dense and diverse community. And so these microbes are, constantly interacting with each other, and those interactions lead to the health benefits, and also the diseases, microbiome-related diseases that can arise. So the microbiome typically exists in a healthy homeostatic state where the microbes are performing beneficial functions for human health. In response to environmental perturbations, the community becomes unbalanced, or the population structure changes and the functions of the community change in response to those disturbances. And that can lead to these feedback loops, which reinforce disease, inflammation, and this state is called dysbiosis. So underlying this kind of balance between a healthy and disease state are the metabolites that the microbiome is producing and degrading. And these metabolites are, being discovered over time and their mechanisms. But here's just a short list of some of the metabolites that have been identified in microbes. So these are metabolites that are produced by the gut microbiome or potentially degraded or transformed. And they have important impacts on human health, disease, nutrition, behavior. And central to all of these functions are the interactions, right? So we really want to understand these interactions because these communities are operating as a unit to impact human health. There's not a single microbe that is operating in isolation. And so we like to think about kind of interventions that we could engineer. And so we've been considering kind of dietary factors that we could manipulate in combination with defined bacterial communities. And the goal would be to design these combinations of nutrients and defined communities to steer microbiomes to desired healthy states and produce metabolites that are beneficial, protect against disease. So I just want to give a little primer on modeling. So as I mentioned, the microbiome is really complex, and we can't just look at microbiome data and try to figure out what's going on. It's really hard. So we use computational models to decipher interactions within microbiomes, predict and design their functions. Some of the challenges are that we need to be able to predict the temporal changes. The system's highly dynamic, as I mentioned. The interactions are complex. They can be pairwise, they could be higher order, where the interactions actually change as a function of context. The growth and metabolism of species are coupled, so they're not independent. And so there are feedback loops that arise between the host and the microbial community, and also even within the microbiome itself. And so another challenge is that machine learning is really powerful, but we want to be able to decipher interactions in these systems and not treat them just as a black box. And so we want to have tools for interpretation and for deciphering these interactions that are highly dynamic and complex. So we use computational models and a variety of different types of computational models depending on the question we're asking. And I'll highlight a couple different approaches today. So the first vignette I wanted to talk about is related to community stability. So we know that if we want to design a microbiome to address some grand challenge facing human society, we need to think about stability as a major function to optimize and manipulate. So we were interested in nutrients and how they shape microbiomes. So we know that dietary fiber has many health benefits, although if you dig into the literature, there are some deleterious outcomes sometimes taking dietary fiber. So it's not always a beneficial nutrient to consume. But for the most part, it's linked to many beneficial effects on human health. But one of our biggest knowledge gaps is that we don't know how to predict how the microbiome responds to these dietary fibers. So we we're interested in kind of distilling the complexity of the microbiome to a 10-member synthetic community, which we designed to represent different metabolic niches. And some of these microbes produce butyrate, which is a beneficial molecule for human health, and kind of span some of the phylogenetic diversity in the gut. And we considered nutrients that vary in chemical complexity, and we quantified the chemical complexity similar to looking at the entropy of the molecule. And we considered inulin, laminar, and xylin, which are three fibers that are present in the human diet. And then we also compared these fibers to their respective sugars in the appropriate ratios, the same ratios that are in the polymer. So we have the respective sugars and comparing directly to the polymer itself. So by comparing these two conditions, we can analyze the effect or the contribution of the glycosidic bonds within the fiber to the assembly and stability of the community. So this project was led by we're still working on it in different directions, by Aaron Ostrom-Loss, who's a postdoc in the lab. And so the first experiment that we did was to look at how these microbes grow in monoculture in response to the fiber. So we just measured their growth as a function of time, and this heat map is quantifying the extent of growth. And so a lighter color indicates that the microbe grew more over a defined time window. And we looked at different concentrations of these fibers and nutrients as well. And so really did a detailed characterization of the monoculture growth response to these different nutrients, either the sugars comprising the fiber or the fiber itself. And we find, maybe not surprisingly, that if we look at the total growth summed across monocultures, so this is just our naive prediction of what we would expect, how the community would grow in response to the fiber, but instead we're looking at monoculture, we see a negative correlation with carbohydrate complexity. So the complexity of the polymer. This is indicating that as you increase the complexity, chemical complexity of these molecules, then we're constraining the growth of these gut bacteria. So we wanted to understand what actually happens in communities. And so we took the 10-member community and we performed an experiment called a serial dilution, where we grow the community for a period of time and then transfer an aliquot of the community to fresh media, so we can follow the community over a longer time scale. And did this over 4 passages total. So it's 3 days, so 72 hours. And what I'm showing here is the community composition measured at the endpoint of this passaging scheme across different concentrations of fibers. And it's complex, as you can imagine. We don't really understand exactly what's happening yet. But one thing we did to kind of simplify and understand what was happening in the community is we distinguished 2 metabolic niches, either responder species grow in monoculture in response to the fiber, or non-responders. These are bacteria that don't respond to the fiber. So over that 72-hour period in monoculture, do they actually display enhanced growth in the presence of the fiber? So we can separate these species into 2 categories. And we can look at kind of the trends in their proportions of responders and non-responders in the community. And this will give us greater insight later. One of the things we actually found was that if we look at the growth response of these bacteria in monoculture, the growth is negatively correlated to the proportion of non-responders. So this means that as we restrict the growth of the communities, the community with fiber, with chemical complexity of the fiber, we promote the growth of bacteria that don't directly grow in response to the fiber in monoculture. So this kind of allows a niche to expand as we restrict the growth of the community. And this was a very strong correlation in our data, so we were really intrigued about what was actually happening. And we also see this as a function of concentration. So both concentration and chemical complexity are allowing the expansion of this unique niche, set of niches that don't depend on fiber. And so we did something quite ambitious, is we took high throughput measurements of microbial communities across all of these different nutrients, and which requires looking at a lot of different communities and a lot of different contexts. And we built a computational model called a generalized Lockable-Terra model. It's a dynamic ecological model that captures interactions between different species in the community. And it represents kind of the growth dynamics of the community and the pairwise interaction. So it doesn't capture higher order interactions. And what we found actually was that as we go from inulin to inulin sugars, we see a balancing of the proportion of positive and negative interactions in the community. So, and this is true also for laminarin and for xylan. So these fibers, chemical complexity, promotes a more balanced proportion of positive and negative interactions within the community. And this in turn affects community assembly and, as I'll talk about next, response to perturbations. So in the theory literature, in ecological theory, a balanced positive and negative interaction network tends to be more stable. This is in theory and simulation with computational models. And so we were really excited to see that fiber kind of promotes a more balanced network. We did a series of experiments to understand how communities respond to perturbations. And just considering the fire example, or treating a microbiome with antibiotics, there is a mass mortality, large mortality, and microbiomes have to reassemble as a community. And so we considered that type of perturbation where we vary the initial proportions of different bacteria in the initial inoculum. And then we perform this passaging experiment and look how the community assembles over a longer time frame. And what we find is that communities grown in complex carbohydrates tend to assemble extremely reproducibly, like they always assemble to the same community. Whereas communities that have greater growth or they're grown in less complex carbohydrates or higher concentrations of carbohydrates, we see a wide range of variation in the assembly of the community. So this tells us that the growth restriction by fibers promotes more reproducible community assembly, whereas when there's a high amount of metabolic activity due to microbial metabolism in less complex carbohydrates or higher concentrations of carbohydrates, then we see a wide variation in the community dynamics. This trend was also recapitulated by our generalized Lotka-Volterra model. It was also recapitulated by a consumer resource model. And so this tells us that when you have more competition between microbes, they tend to have more variation in their assembly dynamics. And this is kind of intuitive, but it was really nice to see that you could recapitulate this with a very simple model of competition. The final kind of test that we did was to challenge our community with invasion. So invasions are relevant in many different contexts, but we can think about this resident community that we have, and then we introduce 26 different bacteria into this community under a wide range of nutrient environments that we've been studying. And on the y-axis here is the change in resident community composition. So that's how much the resident community, the 10-member community, changed in response to this invasion perturbation over a longer time window, which was 3 or 4 days. Actually, maybe longer, 8 or 6 days. And so what we found is that carbohydrates that are more complex stabilize the resident community in response to invasion. So whereas carbohydrates that are less complex or even higher concentrations, they actually promote a larger change in the community composition, the resident community composition. So this is another demonstration that growth restriction of communities can be stabilizing and enhance the resistance of communities to invasion. The final thing I'll mention here is a little bit more of a complicated experiment where we actually used a design of the community to vary the degree of niche overlap between different species. And we find that if you either minimize the niche overlap or you maximize the niche overlap, you get an enhancement of community stability in response to invasion. At intermediate levels, there is a greater degree of change. So the community is more sensitive to perturbations. So this is a little bit intuitive if you think about species occupying orthogonal niches, and they will just continue to grow on the nutrients that's present in the environment in response to invasion, and the community composition will change to a smaller degree. And then if you have communities that have a lot of competition, they're able to compete away all those nutrients so that invaders can't establish. So these are two strategies for maximizing the stability of communities in response to invasion. So I'm going to kind of switch gears in the remaining time that I have to talk about metabolism. So I mentioned that stability is one of our major goals, but also we want to be able to design new metabolisms, optimize existing metabolisms for our benefit. And we've been really interested in the metabolism of these gut communities, notably butyrate, which is a fermentation end product that's present in millimolar quantities in the human gut, but it varies widely across individuals. And we don't have any strategies currently to manipulate butyrate levels. So butyrate promotes homeostasis in the gut. It's linked to a variety of different beneficial health outcomes. And so we were interested in how to design tailored metabolisms. How do we actually design a community from the bottom up with a target metabolic profile? And so this project was led by a postdoc in the lab, Dr. Ryan Clark, and we considered a 25-member synthetic human gut microbiome and used a two-stage modeling approach where we can model the growth dynamics of the community and then map the species abundance at a specific time point to the concentration of metabolites. And so we use a statistical model to take the concentrations of those species and map it to the concentration of metabolite over a time window. And we use kind of an engineering inspired approach called a design test learn cycle, where we can take some measurements in the lab. We can't explore all possible communities. It's over 26 million combinations of different microbial communities that we could assemble in the lab, which is currently not feasible experimentally. And so we selected communities using kind of a model guided strategy. So we'd go into the lab, we'd take some measurements, we build a model, we'd use the model to guide the next experimental design. And we can do this iteratively until we've arrived at a model that gives us new biological insights and that we can use for design. This kind of function, butyrate, we can visualize on a landscape. And so this scatter plot is showing all the possible communities containing all 5 butyrate producers. We have 5 butyrate producers in our community. So every data point represents a different community. And on the x-axis is the predicted total butyrate producer abundance within our community. And the y-axis is the predicted butyrate concentration. So our computational model, when it was informed by some experimental data, predicted kind of a bimodality in the landscape, where we see certain communities having higher butyrate than others, and it was linked to the presence and absence of a bacteria called D-sulfovibrio piger. So D. piger is a hydrogen sulfide producer and has a very unique metabolic niche in the community. So through this iterative process, we were able to design the community, design communities that have 10 or greater species. And we identified that when we tested our model's prediction, that D. piger consistently in our data, and not just in our model, had a reduction in butyrate concentration. So this bacteria, through its production of hydrogen sulfide, is inhibiting butyrate. And we found, we demonstrated this with experiments I won't get into, but hydrogen sulfide is a major inhibitor of butyrate production in our system. So our model was able to forecast the role of this species, D-sulfovibrio piger. And so we, through this iterative process, tested to see whether our model could accurately predict butyrate concentration across a wide range of community contexts, and found that the model was quite accurate. But there was one thing that was necessary, which is that we needed information from high richness communities in order to actually accurately predict the concentrations of butyrate in our system. So this suggests that there are higher order interactions that are shaping important metabolic functions like butyrate. And our model actually fails to capture these higher order interactions because everything is based on a pairwise model. Another thing I wanted to point out in the data was that we, you would naively think, oh, let's mix together all 5 butyrate producers and we'll get the maximum butyrate production because the microbes are actually not competing over any other nutrients. And so that's what's shown in the data point here, this 5-member community when we mix them together. And actually, this 5-member community produces a reasonable amount of butyrate, but by engineering the interactions in the community where the species actually don't produce butyrate, but they're just competing with those butyrate producers, we can get a 45% increase in butyrate production. So this shows that microbial interactions can be engineered to enhance a specialized metabolic function like butyrate in our system. And actually the 25-member community has one of the lowest butyrate concentrations. So we definitely don't want to simply just mix together these bacteria and expect them to produce high levels of butyrate. There's a deeper understanding of the interactions that's needed. So through a series of experiments, we deciphered kind of the ecological and molecular mechanisms that is shaping butyrate production in our system. We identified key molecules such as pH and resource competition over the sugars that promotes butyrate production. And we also identified kind of 2 modes of butyrate production where there's a switch in the metabolic pathway flux as resource competition increases. So resource competition actually will enhance butyrate production. And this kind of functions as an AND gate in a way. So if you have a high enough pH and strong enough competitors, then we get really high butyrate production. So there are these higher order effects that are linked to molecular factors in the environment that's promoting specific types of metabolisms that we can exploit. I'm not sure, I'm not keeping track of time, but oh, 2 minutes. Okay, so I'll quickly mention this. So our results showed that higher order interactions were really important for butyrate production. And so we wanted to go further to not just predict butyrate, but also predict other fermentation end products such as acetate, lactate, and succinate, which are also linked to a variety of different host physiology and different disease states. And there's one limitation with the models we've used is that they don't capture these higher order interactions. They are predictive. We're able to decipher meaningful interactions. But we were interested in considering more flexible models like recurrent neural networks. And so these models can actually learn really complex interactions between different species in the community. They can learn feedbacks between growth and metabolite production. And they can capture temporal changes in the system. And we also have tools for interpretation. So it's harder, but we can decipher meaningful interactions in these models. So this project was it was a collaborative project led by Dr. Ryan Clark and another postdoc at the University of Michigan. And so we took a similar approach where we have this 25-member community and we can vary the presence, absence of different species and look at now all 4 metabolites that these communities are producing and use our machine learning based approach to predict the dynamics and function of the community. This actually was published already in eLife, so it's out now. So this is a 4-dimensional space. So our model is much more efficient computationally. So we can train it really much faster and we can make predictions much faster than our differential equation statistical model framework. And so here I'm showing all possible communities that are greater than 10 species. And every data point represents a community. And this model was informed by our initial data set. So it has some information about the system. But then we use the model's predictions to select either communities that had extreme metabolic profiles, so they either had maximizing or minimizing the metabolite concentrations, or we're kind of uniformly sampling the space just to challenge our model. And the model is particularly challenged in the corner cases. We call these corner cases, which are the extreme metabolite production, like maximizing and minimizing of all of these 4 metabolites. And so we wanted to challenge our model, and we designed an experiment. We carried it out and we tested the model's predictive capabilities on this new set of communities. This is again using a 25-member synthetic human gut microbiome. And we found that the long short-term memory model, which is this recurrent neural network model, could accurately predict the majority of variables in our system and outperformed many other models that we tested, including random forest and feed forward network, and our hybrid model, which was the differential equation statistical model framework. So this model has greater flexibility. It's able to generalize better and actually capture some of the extreme behaviors that we were able to predict. And we use this model, even though it's considered a black box model, and it is, to decipher both microbe metabolite interactions and microbe-microbe interactions. So now we can look directly at these interactions. Every community has different interactions. So it's very context dependent. So we can take an average across all possible communities, and that's what's shown in this network. So each edge represents either a positive or negative interaction between two variables in our system. And we found that actually Bacteroides, which are highly abundant species, both in the human gut and in our communities, they tend to drive the community growth dynamics, but they don't uniquely contribute to these metabolite profiles. Whereas other species like the firmicutes and the proteobacteria and actinobacteria, they have a very unique contribution to these 4 metabolites that we looked at. Of course, we're not looking at the whole picture of all the different metabolites yet, but that's future direction. So I think I will kind of wrap up here because I think I'm out of time, but we also looked at kind of the temporal evolution of metabolite production, and our model was able to accurately predict community dynamics and metabolite dynamics. And one of the kind of general principles that we took away from this is that communities tend towards typical attractor states. You pick a random community, it tends to have very similar behaviors. In order to actually identify communities with unique or dissimilar behavior, say optimizing the production of a target metabolite, we really have to use computational models to guide us to those particular communities because they're very rare in the landscape. And so that's one of kind of the general principles that we found. I won't go into the details of this minimal spanning tree, but that's the general conclusion from that. So in summary, glycosidic bonds and fibers, they restrict microbial growth, they balance positive and negative interactions, and they reduce the sensitivity of communities to perturbations. Microbial interactions can be engineered to design and optimize functions. And we have to use, or we should use, this coupled strategy of high throughput experimentation and modeling, because it's very powerful for identifying these very unique communities with desired functions. And I think ultimately, if we gain a more detailed and quantitative and predictive understanding of microbial communities, we can exploit this ecology to our benefit. And I think this has a wide range of applications beyond human health, agriculture, and beyond. So I think this kind of quantitative understanding will enable us to exploit these interactions in new ways. I'd like to thank my group is fantastic to work with, our collaborators. We're also recruiting for a couple projects, studying secondary bile acid transformations in microbial communities and gut communities, and then also a more environmental project designing anti-mold consortia. So if you're interested in PhD or postdoc or something else, just let me know. We are recruiting for several projects, and I'd be happy to take any questions.
All right, we have time for a quick question before lunch. Yes.
Yeah, sure.
I was wondering if you focus on production. Maybe this is baked into just which part of the pathway you're looking at, but do you think, how do you think your conclusions would change if you were trying to do something for bioremediation here to engineer a community to consume something?
Yeah, that's a great question. We are looking at degradation of toxic metabolites. We're using a different strategy where we're engineering a metabolic pathway for degradation of this toxic metabolite. But we haven't looked at community interactions and how they contribute to degradation. I would expect, like, usually these degradation pathways are specialized pathways, so they're in very unique microbes. So any interactions that promote the growth of those microbes or potentially enhance the metabolic function or the enzymatic function or flux through that pathway would allow increases in the degradation of these compounds. So I think they'd be very similar approaches that we could use for studying degradation as we do production? But that's a great question. I'll think about it more. Yeah.
All right, I would say we defer all other questions over lunch, which will be served at IGB on the terrace on the 3rd floor. And let's thank Ophelia again for the excellent talk.